from the biomolecules. Firstly, we have covered up what are biomolecules. Those are carbon compounds in the living organism, and then we have analyzed the elemental analysis. Elemental analysis, where we have observed the concentration or the percentage of the elements, the abundance of the elements that are present in human body and in earth crust. Okay. And then we have moved on to, where is the content that I have written? Just a second, ma. No issues, no problem. So, we have learnt that carbon, hydrogen and oxygen, these are the elements which are relatively abundant in human body. And then we have learnt about chemical analysis. Chemical analysis, where we have taken one living tissue, be it plant tissue or animal tissue, we have grinded up with what chemical? Trichloroacetic acid using mortar and pestle. And then we have drained it with cheesecloth. We have got a solid liquid content. We have taken that in the test tube. Performing centrifugation, we have got two fractions. What are those two fractions? Filtrate and retentate. In filtrate is also said as acid soluble pool. Retentate is also known as acid insoluble pool. In acid insoluble pool, we have observed all the biomacromolecules. Biomacromolecules include lipids, proteins, nucleic acids, carbohydrates. Okay. Filtrate or acid soluble pool consists of all the cellular constituents, inorganic compounds and elements. Okay. Where are secondary metabolites observed in which pool? In which pool do you observe secondary metabolites and primary metabolites? Secondary metabolites only. Acid soluble pool, you will observe the secondary metabolites. You will get the secondary metabolites in acid soluble pool. Primary metabolites like polysaccharides might be proteins. These will be obtained in acid insoluble pool. Understood, ma? What will be the average molecular weight of the molecules that are obtained in acid insoluble pool? About 1000 Daltons. What is the range of molecular weight of those components obtained in acid soluble pool? 18 to 800 Daltons. Are lipids strictly macromolecules? No. Then why they are placed in acid insoluble pool? Anyone. Yeah, it is insoluble in water. It forms micelles due to disintegration of cell membrane, and its molecular weight is less than 1000 Daltons. Okay, these three are the major reasons to say that lipids are not strictly macromolecules. Understood, everyone? Yeah. And then we have looked into the biomacromolecules. We have first analyzed the primary and secondary metabolites. What are the metabolites, ma? Precursors, intermediates or end products of any biosynthetic pathway. Tell me few examples of primary metabolites. Carbohydrates, lipids, proteins. Yeah. Secondary metabolites? Alkaloids, flavonoids, rubber, resins, colored pigments. These are the examples of secondary metabolites. And the table which is given in your NCERT for metabolites is very important. Secondary metabolites, different types of secondary metabolites you have learned with a mnemonic. Pigments include? Alkaloids include morphine and codeine. Terpenoids include monoterpenes and triterpenes. Essential oils include? Toxins include? Resin. Lectins include? Concanavaline A. Drugs include? Vinblastine and coulsonine. Polymeric substances? Rubber, gums and cellulose. Okay. These are different secondary metabolites that you have to remember, and fix it in your mind. They are again obtained in acid soluble pool. Yesterday itself I have told you, except those biomacromolecules. Okay. Nucleic acids, polysaccharides, proteins and lipids. Everything else which are present in a cell are obtained in acid soluble pool itself. Then about biomacromolecules, we are done with carbohydrates, which are also addressed as saccharides. Classification of carbohydrates include monosaccharides, oligosaccharides and polysaccharides. Can anybody, only one person, tell me the examples of monosaccharides from this row? Maltose, good. Maltose, lactose and I asked about monosaccharides. Glucose, no problem. Glucose, fructose, galactose, mannose. These are the examples of monosaccharides. Glucose, fructose, mannose, galactose and hexoses. What are the examples of disaccharides? Now you tell me. Maltose, sucrose, lactose. What are the examples of trisaccharides? Raffinose, tetrasaccharide, stachyose. Okay. What is addressed as sweetest sugar? Fructose. Okay. Universal sugar? Glucose. What are addressed as monosaccharides? Monosaccharides 3 to 7 carbon units. Are they known as reducing sugars? Yes. Why they are known as reducing sugars? Any one person. Yeah, they have free functional group. If they have free functional group, why are they known as reducing sugars? Because they help in the bond formation between two monosaccharides, thereby they get reduced. Okay, fine. And it also reacts to a reducing agent. What is that? Tollens reagent. Fine, right? Maltose is a disaccharide. What are the monosaccharide units in the maltose? Glucose. What bond is formed? Alpha 1,4 glycosidic bond. The bond between what are the units that are present in the lactose? Monosaccharide units. Galactose and glucose. Alpha 1,4 glycosidic bond. Sucrose has two monosaccharide units. What are they? Glucose and fructose. Alpha 1,4 glycosidic bonds. The bond is in between first and fourth carbon of the two monosaccharides. Understood about disaccharides? Oligosaccharides. Oligosaccharides are primarily formed by condensation of 2 to 9 carbon units. Then about polysaccharides. What are the two types of polysaccharides you have learned? Homopolysaccharides and heteropolysaccharides. Homopolysaccharides examples? Cellulose. Anyone. Homopolysaccharides examples? Starch, chitin, glycogen, cellulose. Heteropolysaccharides? Okay. So, cellulose is a homopolymer, means what? It has only one type of monosaccharide to form that polysaccharide chain. Fine, right? What is that one monosaccharide? Glucose. What bonds are formed between two glucose units? Beta 1,4 glycosidic bonds. Okay, to form a chain of cellulose, what are the monosaccharides used? Beta 1,4 glycosidic bonds. And where do you find cellulose? Plant cell wall, pulp of plant, cotton fibers, paper pulp. Okay, these are where you find the cellulose. Next is starch. Starch is also a homopolymer which is made up of alpha D glucose, but the chains formed are different. What are they? Amylose and amylopectin. Amylose is formed by what? Straight chain alpha 1,4 glycosidic bonds. Amylose will have alpha 1,4 glycosidic. It is a straight chain. Do you find any branched chains there? No. Whereas amylopectin will have branched chain. It has two types of linkages, alpha 1,4 glycosidic linkages, alpha 1,6 glycosidic linkages. Okay. 80% of starch will have amylose or amylopectin. And does starch give a positive test for iodine? Yes, it gives blue color. Cellulose doesn't give. Why? Because it doesn't have complex helices. Next is glycogen, which is the storage food material in animal cells. Glycogen is another polymer which has both alpha 1,4 glycosidic linkages and alpha 1,6. The image which is given in your NCERT, wherein the right end is said as left end is non-reducing end. This reducing end will have free aldehyde and ketone groups. In the diagram it is only aldehyde groups. Okay, it has free aldehyde groups where it can connect with the other monosaccharides. Whereas the left end is non-reducing end. Fine, right? It is non-reducing end. It doesn't have any free aldehyde or ketone groups. Clear, right now? And then, very good. Next, chitin. Chitin is also known as fungal cellulose. It is present in fungal cell wall and exoskeleton of arthropods. And what are the glucose units that are present in chitin? NAG units. N-acetyl glucosamine. Okay. Then about inulin. Inulin is a polymer of fructose. Where do you find that is not at all required. But remember one thing in human physiology, that inulin clearance is equal to glomerular filtration rate. Okay. Inulin is not absorbed by the human body, thereby inulin clearance is equal to glomerular filtration rate. And then about proteins. Proteins, do you find any homopolymers? No, there is no possibility. It is a heteropolymer of what? Amino acids. Because each amino acid will have an alpha carbon at the center, to which carboxyl group is present, amino group is present, hydrogen is present. This is common for all the amino acids. The difference comes from R group, which is the functional group. 3 examples given in your NCERT. If R group is H, you address that as glycine. If R group is methyl group, then it is alanine. If R group is hydroxymethyl group, it is serine. Okay. But then you have to remember all the different types of amino acids. Basic classification on the polarity, okay, where amino acids are classified into polar amino acids and non-polar amino acids. Polar amino acids we again have positively charged, negatively charged. Negatively charged will have less pH, okay. Negatively charged amino acids include aspartic acid and glutamic acid. Positively charged polar amino acids again includes what? Lysine, arginine, histidine, serine, threonine, aspargine, glutamine and cysteine. Okay. Next is hydrophobic non-polar amino acids. We have aliphatic, aromatic. Aromatic examples: phenylalanine, tryptophan, tyrosine. Yeah. Essential, non-essential amino acids. To remember the essential amino acids, I have given you a mnemonic, right? PVT. Total 9 essential amino acids: phenylalanine, valine, tryptophan, tyrosine, isoleucine, methionine, histidine, alanine, leucine and lysine. Fine, right? These are all essential amino acids where you will take it from the nutrition or diet that you take. Okay. And then comes the zwitterionic structure. What is zwitterion? The amino acid at the isoelectric point, that is at neutral pH. It has both acidic charge and basic charge. If you even more reduce the pH, that turns to what form? Cationic form. Okay, where positive charge is at the amino group. If you increase the pH, it turns to anion. Okay, and the negative charge is at carboxyl group. Understood? Zwitterion is very important. Considering with the biochemistry of your chemistry subject also, you have one biomolecules chapter, right? There also zwitterion concept will be there. It can be asked anywhere. We have learned that glycosidic bond is formed between 2 monosaccharide units. Fine. What is the bond that is formed between 2 amino acids? It is formed between carboxyl group of one amino acid with amino group of the next amino acid. Okay. By dehydration again, the water molecule will be lost. Okay. See, this is how the peptide bond is formed. Fine. And based on the level of organization of different amino acids, we have 4 levels of protein structures: primary structure of proteins, secondary, tertiary and quaternary structure of proteins. Amongst these structures, tell me which structure will give you the positional information? Primary structure, wherein it is just formed by a peptide bond. At first amino acid will have what free terminal end? N terminal end. Last amino acid will have? Where do you find alpha helix and beta pleated? Hollow wool-like structure is formed in three-dimensional structure. What are the bonds involved in secondary structure? Hydrogen bonds. Hydrogen bonds in alpha helix, it is formed between 4 down amino acids, and hydrogen bonds between preceding amino acids is formed by beta sheets, beta plated sheets. And then quaternary structure will have subunits, right? 4 subunits are formed. Best example is hemoglobin, 2 alpha subunits and 2 beta subunits. One more question. What are the bonds involved in tertiary formation of tertiary structure? Dipole-dipole interactions, ion interactions, hydrophobic interactions. These are the bonds which are involved in tertiary structure. You are clear with the class? These are the diversified proteins that are present. Okay. Collagen, which forms intercellular ground substance. Trypsin is the enzyme. Insulin, hormone. Antibody fights against infections. Receptor, sensory reception. GLUT4 enables glucose transport into the cells. Fine. What is the abundant protein in the biosphere? Rubisco. What is the protein that is abundantly present in the animal world? Next is nucleic acids. The basic structure of nucleic acid, the formation of nucleic acid is nucleotide. So, nucleic acid is also addressed as polynucleotide chain. Single nucleotide is composed of sugar, nitrogen base, phosphate group. If it is DNA, what sugar is present? If it is RNA? To form a single nucleotide, what are the bonds involved? Single N-glycosidic bond, phosphoester bond. N-glycosidic bond is formed between which carbon? First carbon of the sugar to N9 of purine and N1 of pyrimidine. And phosphoester bond is formed between C5 of glucose with phosphate group of phosphoric acid. Understood? That phosphate itself is giving the negative charge to DNA. Understood, ma? I hope there is no repetition required for different types of nucleotides that we have discussed yesterday. You are clear on that, right? Yeah, these are different nitrogen bases. Now, how a polynucleotide chain is formed? Firstly, a single nucleotide C1, C2, C3, C4 and C5. Okay. Here nitrogen base is attached, yes or no? And here phosphate group is attached. This forms out a single nucleotide. At third carbon and second carbon, what are present? OH group is present. Second carbon, OH will not be there if it is DNA. Here the OH which is present to this phosphate group is adjoined, which is connected to fifth carbon of the succeeding nucleotide, like this. Got it, ma? So, here to form a single nucleotide, here what bond is involved? Between first carbon and nitrogen base. What bond is this, ma? N. Don't get confused. See, clearly you observe. This is clear, no? Between C1 and nitrogen base. If nitrogen base is purine, it is connecting to N9 of purine. If it is pyrimidine, it is N1 to pyrimidine. What bond? N-glycosidic bond. Here what bond is involved? Phosphoester bond. and here OH group which is present at third carbon, 2 phosphate group which is present at fifth carbon of the succeeding nucleotide. This will be connected via phosphodiester bond. Okay. This is how a single nucleotide chain is formed. You will say single stranded DNA, single stranded RNA, that single stranded RNA is formed by these bonds. Okay. Single stranded RNA between two nucleotides. Two nucleotides are connected by what bond? Phosphodiester bond. Fine, right? This forms phosphodiester bond. So, to form a single chain of DNA or single chain of RNA, what are the bonds included? N-glycosidic bond, phosphoester bond, phosphodiester bond. Clear, right? Yeah. Excuse. Understood, everyone? Yeah. Now, to form a double-stranded DNA, what happens is you have to understand, you have to get to know about the double helical model of the DNA. This is clear, no? How a polynucleotide chain is formed. Two nucleotides are connected by phosphodiester bond. And then here you observe, at 5' end, what is present? Free phosphate group is present. At 3' end, OH group is present, which is nothing but C. At the top, 5' end, what is there? Phosphate group is there. Here at 3' end, what is present? OH group will be present. Same like in a protein structure. First amino acid will have free N-terminal end. Last will be having free C-terminal end. Here at 5' end, at fifth carbon, at first position, phosphate group is present. At 3 terminal end, what is present? OH group is present. So, they run anti-parallel to each other in a double-stranded DNA. One from 5' to 3' and then from 3' to 5' like this. Fine. So, here when a double helical structure is formed, the backbone of the double helical DNA will be sugar phosphate group. At the center, nitrogen bases will be present. These nitrogen bases are connected by hydrogen bonds. Okay. Adenine is connected to guanine by 2 hydrogen bonds, cytosine to thymine via 3 hydrogen bonds. Sorry. Adenine to thymine, guanine to cytosine. Now it is right, no? Sorry. So, here in the double helical model of DNA, we will learn about B-form DNA. There are many forms of DNA. In molecular basis, we will learn comparative analysis between A form, B form and Z form. Right away, we will discuss the stable form of DNA, that is B-form DNA. It is said that one complete turn of the DNA, one complete turn means from here to here, one complete turn of the DNA, the length of the DNA is 3.4 nanometers or 34 angstroms. Fine. Here you can, just a second. Here you can also look into the comparative analysis once. We have enough time. Yeah. In this, what you have to focus is, first, how the double stranded DNA is folded. A type DNA is right-handed helix, B type DNA is also right-handed, Z DNA is left-handed DNA. Number of base pairs that are present are 11 base pairs, here 10 base pairs, 10 base pairs you have to consider, and here 12 base pairs. Major groove means this is addressed as major groove, the gap which is formed because of the turns, and this is addressed as minor groove. Major group shapes are important. It is narrow and deep in A type DNA, B type DNA it is wide and deep, Z flat or absent. Minor groove is wide and shallow in A type, narrow and deep in B type, narrow and deep in Z type DNA as well. Okay. And the most stable DNA amongst these three is B type DNA, which occurs in all the types of cells. Okay. If these points you can note it enough, how major groove and minor groove are formed, what are the number of base pairs of A, B and Z DNA, in what fashion they are folded, 1, 2, 3, 4, and which is the most stable DNA. If you are done, just let me know, ma. Done, right? Yeah. Just look into the Chargaff's rule which are being proposed by looking into the double helical model of DNA. It is Erwin Chargaff in the year 1950. He has said that, see, this will be with you only, you can copy later as well. Okay, we are running out of the syllabus. Erwin Chargaff in the year 1950, he concluded that any DNA will be having equal amount of purines and pyrimidines. The amount of adenine is always equal to the amount of thymine, and amount of guanine is always equal to the amount of cytosine. Then comes the concept of base ratio, that is adenine plus thymine by guanine plus cytosine will vary from one species to other. Usually in prokaryotes that will be less than 1, in eukaryotes it will be more than 1. If you observe, In prokaryotes, the base ratio, prokaryotes like E. coli, the base ratio is around 0.9. In eukaryotes, like if you have to consider human cell, it is 1.3. The deoxyribose sugar and phosphate components will also occur in equal proportions. These are the points that are derived from double helical model of DNA by Erwin Chargaff. Please take down. If you learn it better here, it will be easy for you in molecular basis. Okay. Done? That examples are not required, but just you know you have to know about the comparison between prokaryote and eukaryote enough. Next, about types of RNA. You have 3 types of RNA in eukaryotes. We have already discussed in cell the unit of life also. If you have to consider this as nucleus, okay, nucleolus. Inside nucleolus, what is synthesized? rRNA. By what polymerase? RNA polymerase 1. In nucleoplasm, tRNA and mRNA are synthesized. mRNA is synthesized by what polymerase? RNA polymerase 2, and tRNA is synthesized by RNA polymerase 3. Okay. So, messenger RNA is of just 5% of the total RNA. What is the major function that it does? It does carrying of the genetic code from DNA to ribosomes. Ribosomal DNA plays structural and enzymatic role in the ribosome. It occupies about 80% of total RNA. tRNA, soluble RNA or adaptive RNA. This transfers amino acid to ribosome during translation. It is of just 10 to 15% of total RNA. Take down quick about the RNA polymerases which are synthesizing different types of RNAs and what is the amount of RNA that is present. Any doubt, ma'am? Done, everyone? Next biomacromolecule about lipids. Lipids are water soluble or water insoluble? Water insoluble. They are not polymers of any monomers, okay? They are not polymers of any monomers. They are formed because of the association of different molecules or globules, okay? They are of three types. Lipids are of three types we will learn. One is simple lipids, second derived lipids, third compound lipids. Okay, simple lipids, derived lipids and compound lipids. First we will learn about simple lipids. They have a fatty acid and a carboxyl group to an R group. Usually lipids general formula, if you have to take out, it is RCOH. To the carboxyl group some functional group will be adjoined. Okay, the R group can be anything. It can be methyl, ethyl or higher number of carbon groups also. That carbon groups can vary from 1 to 16 to 1 to 20 also. For example, palmitic acid, if you have to consider, palmitic acid will have 16 carbons including carboxyl carbon. Arachidonic acid will have 20 carbons including carboxyl carbon. Okay, these are the examples you have to learn. Okay, first simple lipids, derived lipids, compound lipids. Okay, lipids are usually water insoluble. What is the general formula and how many carbons does palmitic acid have including carboxyl carbon? How many carbons does arachidonic acid have including carboxyl carbon? Then simple lipids are of two types. We have neutral or true fats and glycerol. These are esters. True fats are nothing but esters of fatty acids with the glycerol. Okay, esters of fatty acids with the glycerol. So they are called as what? Glycerides. For example, glycerol if you take, it has 3 carbons, each is bearing one hydroxyl group. Each carbon is having one hydroxyl group. As it has 3 hydroxyl group, it is noted as trihydroxypropane. Glycerol is the common name, IUPAC name will be trihydroxypropane. This is the example of one type of glyceride. Okay. If only 2 OH groups are present, that is dihydroxyethane. And only CH2OH to monoglyceride, diglyceride, triglyceride. Next, fat, you can copy it later, listen first, okay, because we have to discuss about enzymes as well. Fatty acids could be saturated or unsaturated. Saturated, they will have only one single bond between carbon units. If 2 bonds or 3 bonds are present, you will call it as unsaturated. Saturated fatty acids examples, only examples you see, palmitic acid, arachidonic acid. These are the examples of saturated fatty acids. These are the common examples given in your NCERT. For saturated, it is palmitic acid and stearic acid. Unsaturated fatty acids, oleic acid and linoleic acid. Linolenic acid. These are the examples of unsaturated fatty acids. Note down the examples. And then this is about, I told you, right, monoglycerides, diglycerides and triglycerides. Number of OH groups that are present with carbon units. Next, based on the melting point, again triglycerides can be fats or oils. You tell me, it is easy to note, right? Which will have high melting point and which will have low? Fats will have melting point and they remain solids at room temperatures. Oils will have low melting point and they remain as liquids or oils at room temperature. Next, about derived lipids. Okay, derived lipids are, the best example of derived lipid is steroids. Best example of steroid is cholesterol. Okay, the steroids do not contain fatty acids, but they are included in lipids because they have fat-like properties. The most common steroid is sterols, example cholesterol. This is most abundant in animal tissues. Food rich in animal fats will contain cholesterol. This is also essential to maintain fluidity of plasma membrane. Cholesterol maintains fluidity of plasma membrane. Done about derived lipids. Immediately when you will recall the derived lipids, you have to remember about cholesterol. It is majorly present in animal cells and it is a major composition to maintain the fluidity of plasma membrane. That's it. Conjugated or compound lipids. We have learnt that lipids will have functional group with carboxyl group. We have learnt about glycerides. Apart from this, if there are any other compounds that are present for the formation of lipids, they are addressed as what? Compound lipids or conjugated lipids. See here, the components that are present for phospholipids, glycolipids, lipoproteins and chromolipids. These are 4 different types of lipids in compound lipids. Phospholipids, glycerol or other alcohol, along with that it also has what? Phosphate group. Okay. It also has choline, which is a nitrogen containing component. Okay. So, what are the extra constituents that are present along with glycerol and fatty acids? Phosphate group and choline. Okay. What are compound or conjugated lipids along with glycerol and fatty acids? If there are any other constituents that are present in the lipids, they are known as conjugated or compound lipids. In phospholipids, what is present? Phosphate group and choline. Major function, this forms cell membrane. Amphipathic molecules, amphipathic are nothing but which has both hydrophilic and hydrophobic groups with both polar heads and non-polar tails. Example is lecithin. Where did you learn about lectin? Secondary metabolite. Example of lectin? Concanvaline A. Lecithin is a major composition of phospholipid. Okay, remember all the time in considering the fluid mosaic model, he will ask you. Okay, lipid is a major composition of plasma membrane. Amongst lipids, which type of lipid is abundantly present? Phospholipid. In phospholipid, what is the major composition? Lecithin. Glycolipids, fatty acid and alcohol. Spingo's sign, it is addressed as glyco. The name itself is saying that the extra component is sugar. Next is lipoprotein. Lipids, along with that it has proteins. This gives the structural components of membranes. It transports lipids into the blood. Next, chromolipids. The name itself is saying it has pigments like carotenoids, etc. These are pigmented lipids. They are involved in coloration and photosynthetic processes as well. Examples include keratin and vitamin A. Understood, ma? Yeah, these are about conjugated lipids. Have an eye on it for 2 minutes and then we will move on to enzymes. These are the images given in your NCERT. See, palmitic acid along with carboxyl carbon, 16 carbons. Lecithin, phospholipid. Okay, it has phosphate group and nitrogen containing component choline. Glycerol, triglyceride also you can call it as, because it has 3 OH groups, trihydroxypropane, triglyceride, and then cholesterol. Fine. Remember the structures carefully, you can get a diagram based question. Next about enzymes. What are enzymes? Basically, what are enzymes? What do you consider an enzyme as? In living organism, it is commonly addressed as biocatalyst. Basically, you understand what is the role of enzyme. It will enhance the speed of reaction or rate of reaction. For example, to form a product, let me take one reaction from your Krebs cycle or any example. Okay, substrate along with enzyme will form enzyme product complex and then it gives out the product. To form a product from substrate, for example, if it is taking 10 minutes in the absence of enzyme, in the presence of enzyme it will take 10 seconds. That is the level of accuracy that enzyme will push up for a reaction. It will enhance the rate of reaction. So, mostly the enzymes will be proteins only, but there are few exceptions. There are nucleic acids which behave like enzymes. For example, ribozyme. Ribozyme is a nucleic acid which is behaving like enzyme. Enzymes are organic catalysts. There are inorganic catalysts also which do not occur in the living cell. Okay. So, both of them are performing same function to enhance the rate of reaction, but the properties of organic catalyst and inorganic catalyst will be different. Organic catalyst or biocatalyst is one and the same. The properties between organic and inorganic catalyst will be different. These are the differences between enzymes and inorganic catalysts. Enzymes are also known as organic catalyst or biocatalyst. Here we go. Enzymes are complex biological macromolecules. They are made up of different proteins, and these are inorganic catalysts. They are usually simple small sized molecules or elements. These catalysts will operate only in living organisms. These are found in non-living systems, that is outside the cells as well. Each enzyme is highly specific to single biochemical reaction. Here a single inorganic catalyst can act on multiple different chemical reactions. Enzymes lose activity after 40 degrees Celsius, they get denatured, enzymes get denatured, but here they are stable even at higher temperatures and pressure, inorganic catalysts. These are the basic differences between organic and inorganic catalysts. But how about those enzymes which are extracted from thermophilic organisms? Thermophilic organisms means those organisms which live in a high temperature conditions. These enzymes from thermophilic organisms, they are thermally stable and they retain their catalytic activity at higher temperatures, like up to 80 to 90 degree Celsius. They make them thermally stable. So, here if you consider the enzyme, that is sucrase, we have to learn the mechanism of enzyme action and then two hypotheses which are proposed to explain the mechanism of enzyme action. Enzyme sucrase, okay, this enzyme when it is adjoined with the substrate, it forms enzyme substrate complex. Sucrase with sucrose, sucrose is the substrate, sucrase is the enzyme. Then it forms enzyme product complex. Okay, the transition from enzyme substrate complex to enzyme product complex is known as transition state, and the energy required to convert from enzyme substrate complex to enzyme product complex is known as transition energy, which is the highest amount of energy. Remember, the highest amount of energy that is required to transit from enzyme substrate complex to enzyme product complex is known as transition energy. The minimal energy required to transfer from enzyme substrate to enzyme product is known as activation energy. Maximum energy required, transition energy. Minimum energy required, activation energy. Okay. Minimum energy will be required in the presence of enzyme. In the absence of enzyme, maximum energy is required, that is transitional energy. Understood the difference between transition and activation? And then products are being released. Products include glucose and fructose. What are the products? Glucose and fructose. Done, right? Now, two hypotheses. One is lock and key hypothesis. Second is induced fit model. Lock and key hypothesis is not widely accepted because lock and key, if you have to suppose, okay, those are mechanical, the structures are not flexible. He has considered this site as active site to which the key is fit into the lock as enzyme. Key is considered as substrate, lock is enzyme, to which substrate is fit into the active site of the enzyme. That is how it is explained in lock and key hypothesis. See, if you observe, substrate enters the active sites of the enzyme, and here enzyme substrate complex is formed, converted to enzyme products, and then products will be released. Induced fit model is nothing but hands to gloves, like they are flexible. Any glove you give, it will fit into the hand. Okay, so that is more applicable to the biological phenomena when compared to lock and key hypothesis, which is static, stable. Okay, this flexible model of induced fit model, which is proposed by Costland, is widely accepted because it is flexible. These are the key differences between lock and key hypothesis and induced fit model. See here, lock and key hypothesis proposed by M.L. Fisher, induced fit model by concept behind this is enzyme's active site is exactly complementary to the substrate shape. Here it is not like that. Enzyme active site changes shape to fit into the substrate. Key fits into the lock, here hand fitting into the glove. This is the analogy, okay, what they have proposed. Next, flexibility of the enzyme. Enzyme structure is rigid and it does not change its shape. Here enzyme structure is flexible. This adjusts to the substrate. Specificity, high specificity due to perfect fit. Here it is moderate due to high specificity. The fit is optimized after interaction. Adaptability to the substrate. This cannot explain enzymes binding to the similar shaped substrates. This will explain the range of similar substrates. Understood, ma? These are the key differences between lock and key hypothesis and induced fit model. Which is widely accepted? What is the energy that is required to convert from enzyme substrate to enzyme product? Maximum energy required. Minimum energy required. Okay. What are the key differences? Any two key differences between organic catalysts and inorganic catalysts? In living organisms, this is outside. Specificity is more for which type of catalyst? Organic catalyst, that is biocatalyst. Okay. Tell me about that nucleic acid which is behaving like the enzyme. Properties of enzymes, which are biocatalysts. They are generally proteinaceous in nature. They may require inorganic or organic substances which act as cofactor. Enzyme alone cannot do its function. Okay. The protein part of the enzyme is known as apoenzyme. Okay. Apoenzyme is the protein part of the enzyme. Apoenzyme is the protein part of the enzyme. Along with apoenzyme, cofactor will be present. This cofactor can be a prosthetic group, it can be a vitamin, it can be coenzyme, it can be anything. So here apoenzyme along with cofactor is addressed as holoenzyme. Protein part is apoenzyme, non-protein part is cofactor. We'll learn about it. Chemical reactions, enzymes do not initiate the reactions but they increase the rate of reaction. Very important in your statement based questions. Enzymes will not initiate the reaction but they will, increase the rate of reaction. Nobody. Next, efficiency. Efficiency is equal to turnover number, which means number of substrate molecules that are converted per minute per enzyme molecule. One enzyme in a minute, how much amount of substrate it is going to convert to the product is known as turnover number. That efficiency is equal to turnover number. Unchanged form, enzymes will remain in unchanged after the reaction. Enzyme specificity, enzymes are highly specific to the substrates. For example, enzyme maltase acts on the maltose, not on the sucrose. Yeah, you can. Continue. Got to know the properties of the enzymes? How you can calculate the rate of reaction? Amount of products formed in a specific time, per unit time. Okay. Number of products formed per unit time will give you the rate of enzymatic reaction. And this enzymatic reaction is influenced by temperature. As you increase the temperature, 10 degrees rise in the temperature will double the reaction. 10 degrees fall in the temperature will halve the reaction. Few more factors will affect the rate of reactions. We will learn this once after noting about the examples which are given in the NCERT. Carbon dioxide and water in the presence of the enzyme carbonic anhydrase. What is the rate of reaction in the presence and absence of the enzyme is given in a comparative analysis. In the absence of any enzyme, this reaction is very slow with about 200 molecules of bicarbonate being formed in a single hour. By using the enzymes carbonic anhydrase, the reaction speeds dramatically with about 6 lakh molecules being formed every second. Okay, the enzymes have accelerated the reaction rate about 10 million times. Got it? Here. What is cited on the x-axis, what is there on the y-axis is very important. X-axis you will see the rate of reaction or progress of the reaction. Over y-axis you will see the potential energy. Here you observe substrate and product that is formed. Substrate here, if the energy of product is less than that of substrate, okay, the reaction is said to be exothermic. If the energy of product is more than that of substrate, the reaction is said to be endothermic. Here you see the energy, substrate is converted to product, the energy required is transition energy, and this is the transition state. This is the amount of energy required in the absence of enzyme, and activation energy with enzyme. Minimal energy required to convert from enzyme substrate complex to enzyme product complex. That's it, you will get to know about the graph, but this graph is very important, okay, which will show about the enzyme mechanism. What is transition state, what is activation energy, what is transition energy, okay. From this graph, how can you identify whether it is an exothermic reaction or endothermic reaction? See, on y-axis you will get to know about the potential energy, right? If the energy of product is less than the energy of substrate, then the reaction is what? Because in the due course of formation of enzyme product, what happens? The energy will be released. If the product is formed, the product which is formed, energy, potential energy is more when compared to the substrate, then it is called as endothermic reaction. Okay, till here you note it. Tomorrow we will be discussing about holoenzyme in detail, and we will practice the complete set of biomolecules questions. Fine, ma? Till here the class is clear, no? We are left over with factors affecting enzymatic activity, and we are left over with cofactors, okay, and objectives of biomolecules. And then we will start a new chapter also tomorrow, plant physiology, that is photosynthesis in higher plants we will be starting, okay.